Thanks for the introduction and great presentation from the OVO guys. I also like the shout out to ProtFlow. I'm Marcus Brown. I'm a graduate student currently at the Technical University of Graz, and I'm part of the Oberdorfer lab, and we just design proteins every day, I would say. Like we really did it a lot over a lot of years. And out of this need to design a lot of proteins came the frustration of working with design tools. And this is really the backstory of how ProtFlow came to be. Before I go into the details, I'd like to say thanks to a lot of people. First, of course, to Adrian, who has been a co-developer for this project from the start, because also he was designing proteins like crazy and he wanted to have something to be able to do it faster as well. And of course, the Oberdorfer lab, great place to work. And all of the contributors and early adopters that we had in our lab, which includes master students and bachelor students, and also other graduate students and postdoc as well. Okay, so with this, I'll go right into something that everyone seems to be observing at this point, and which also seems to be quite obvious from the number of people that have joined the meeting today. It's really surprising that more than 100 people wanted to join in the beginning, or at least to me. Very happy about that. The thing is that protein design is a growing field, and compared to, let's say, 7 years ago maybe, or 10 years ago, where you could do all of your design using Rosetta and Rosetta scripts, today there are a lot of tools around that you kind of need to use and able to just be at the state of the art when you want to make new proteins. And all of this tool landscape kind of gives protein designers real bad headaches. These are all of the problems that we all encounter, starting by having to install all of the tools in the first place. And even worse, when you need to train a new student, you need to explain to your student how to install all of these tools. And every time a new student is coming in, you need to repeat this process. The second thing is, if you want to design something, ideally you want to have this, whatever you're designing, you want to have it automated as quickly as possible, so you can test different design parameters to really squeeze out the best designs out of your pipeline. And this automation takes a lot of work because all of these different tools have different inputs and outputs, and all of this is really a lot of file handling. The next thing is related. Again, once you have your pipeline automated, you really want to analyze which of your design parameters are predicted for success. You need to have a lot of data tracking. You need to keep track of where all of your protein output files are. You keep track of all of your scores, and you need to be able to link them up together. And the last thing is, once you've finished your script and you're finally ready to publish because you get great experimental results, you also need to share your script with other people that need to be able to run it. So in this case, it doesn't count to say the script works on my machine. You really need to be able to easily share your automated script to other people, and they need to be able to run on their systems. And out of these 4 problems that, let's say, we identified while working with designing proteins, out of this, we developed ProtFlow, which is a Python library. So it's for pure Python, to solve the automation, the data tracking, and the portability problems. We didn't try to solve the installation problem yet, because this really changes with tools over time very quickly. Especially if you want to stay updated on the latest versions of every tool. So this was, for now, too much work for us, but we will be open for inputs. Okay. Now, seeing that we claim that ProtFlow solves all of these things, the question is actually, how does this look like in practice? And so I prepared a little bit of a demonstration in how it can look like if you use ProtFlow to design protein. As I said, this is a Python package that you can use either in a regular Python script or also in Jupyter notebooks. And because it's a Python package, we need to import all of the things from the package that we want to use. So in this case, it's a lot of objects, job starters, and tools that we are going to use. So I will be running this. And then the next step is to set up a workspace, which includes setting up the tools that will be running the jobs. This can be seen as a scheduler in Bobo, where you can use a local job starter to run on local machines, or a Slurm job starter to run SBatch array jobs. And the second step, as always, is you need some object that represents the proteins that you're working with. And in our case, because we want to implement this kind of rudimentary diffusion, design, prediction, and analysis pipeline that I showed before, we are starting with a process object that doesn't have any input for now. And directly in the next cell, we can start diffusing monomers. How this works when you want to run a tool is you need to set up the tool that you want to run, which is a runner object. And in order to start this, you give it a JobStarter, and then there is always this run call that you can use that will start sending these jobs. And when I do this now, you can see it executes immediately. So to those people in the audience that use RF diffusion a lot, they will say, what is this? This is too fast. This is because I ran all of this demo before the presentation to be sure that everything works. And if in ProtFlow, you already ran a tool and outputs are already in your workspace, then it will read these outputs and directly read the score files into your poses object. Maybe one or two things important to mention at this point is that my object that I created, these proteins, they have a working directory, which can be either a full path or a relative path, in this case here. And then when I run a tool, every run call needs to have a prefix, which will then be also the directory where all of the outputs of this tool will be. So all of the outputs will be expected to use monomers and then slash for a new directory diffusion. And of course, I have access to all of the outputs instantaneously directly in this Jupyter notebook because my proteins object, this poses object, has a pandas data frame inside that stores and keeps track of all the proteins that you ever loaded, and also of the history of all of the tools that you ran on this protein. You will see this also later when we run more tools. And so in this case, after diffusion, for example, I decided to show the diffusion PLDDTs for this monomer generation, and also the location. So this is another benefit that ProtFlow stores all of the locations of the files that you generate over your design trajectory, which makes it really easy to look things up in the end. Okay, and once we have designed our protein backbones using RF diffusion, we immediately want to generate sequences for them. In this case, we use the ligand MPNN runner, which as before with RF diffusion, we set up the runner, and then we immediately start the run call to execute it. And here I'll share another detail is when you run protein design tools as runners, you can always also add in options, how you would add the options in the command line to the tool that you want to run. So for ligand MPNN, if I were running it in the command line, I would just add this little string to say run at this temperature and don't use any cysteines for design. And this really allows you to customize your tool calls to any of the options, basically, or almost all of the options that the tool is allowing. And you also have the ability to give every single protein in your poses object, so in this data frame, its own different options. So for example, for every protein, if some residues that you want to design are all on different positions, you can just save that information in your poses data frame and then feed it as pose options into the model. Okay, once we run ligand MPNN, I immediately display the output here, where the output of protein MPNN was to design 3 sequences for every backbone that we had from RF diffusion. So in total, we have now 15 rows, where every row is one protein, and every row inherited the scores of the previous tools as well. So here we can see that the diffusion PLDDT from the previous RF diffusion is saved, same as with all of the locations, but I'm just not showing these columns here. And of course, from MPNN, the sequence confidence is also stored in the data frame. Then, because tool calling is not the only thing that you want to do when you set up a design pipeline, I'm sharing one of the utilities that we also implemented for ProtFlow, which you really do commonly, which is filtering your sequences based on particular scores and maybe grouping this filter by some group. So in this case, for example, we made 3 sequences for each backbone, And now we want to take the best sequence of these 3 sequences of each backbone and only predict using AlphaFold later the best of this sequence. So this is what we are doing here with this filter call. Now you can see we filtered from 15 to 5 sequences. And when we display the protein object now, you can see it only has 5 rows. And for every unique RF diffusion output description, we only have 1 sequence in here. Okay, and then to continue in our pipeline, next step is to run ColabFold. Again, it reads the pre-computed outputs immediately, which is why it executes so fast. And now we also have AlphaFold PLDTs for the proteins that we generated. And something that catches my eye when I look at these results is that one PLDT is around 80 and the rest is 98 or almost 98, which is really, really high for a protein structure. So maybe now I really want to know what's going on with these structures. So the next thing that I do is I can also run Rosetta. What we did for ProtFlow is we try to implement a version of a runner that can run any executable that is within the Rosetta software suite. So when you execute this run call, you also add a Rosetta application that you actually run, in our case, Rosetta scripts, because we are executing a Rosetta script that is doing FASTA. And then in the options, we can just add this Rosetta script as command line options, how we would usually do. Running this and then displaying, now we can see we also have the classic Rosetta total score, which is the score that the Rosetta energy function gives you. And similar as before, here we have a very low energy, or not low, but a very bad energy. And for some of the proteins, around 100. Okay, and now when it's finally time to...
Marcus, sorry to interrupt, just a quick question. I see you're using the same pose as working directory. So you've got the generated backbones and then, things from ColabFold. Those are all in the same directory, or how do you pick up just the latest ones?
Do you mean how the latest poses are picked out?
Yeah, so that it's not returning like the ones from the previous.
Yeah. So ProtFlow keeps track of what was the last tool that you ran. And when you supply your poses to a runner, then it automatically uses these, like this column in your data frame as an input. You can actually also change this by hand, although we recommend being very careful with that because we can run into bugs. But it's technically possible. You could run tools on any of the columns that contain any files or proteins that your tools accept in principle. Does this answer your question?
Thanks.
Okay, wonderful. So then as a last thing, as Ayadria was asking, where are the proteins actually? Because ProtFlow stores the locations of the files that were generated by every tool in the data frame, we can select those that we want to look at. And here in this case, I printed a pymol command so that I can open all of them because they are absolute paths. I can just copy and paste this in my terminal. And now the terminal is opening the proteins, and it shows me, of course, RF diffusion generated extended helices because we didn't do diffusion with any monomer radius of gyration potential. And finding this out, we could now just rerun the entire pipeline. Back to RF diffusion, adding another command line argument, monomer radius of gyration potential, and maybe going into a different working directory and immediately have, because we don't need to rerun any code, to rewrite any code, we can immediately see the outputs of that as well. Okay. This is how you can do this in a JobStarter notebook. This was not how we initially designed ProtFlow to work. Initially, this was meant to be run from Python scripts. And so in this case, I cleaned up every tool call that we had here and combined them into a short Python script that does now exactly what we said. And you can see that now in 38 lines, we have this tool chain of diffusion, ligand NPNN, filtering, ColabFold, and then Rosetta afterwards. And yeah, it's really designed to be able to quickly test out the ideas that you have to design the proteins that you want. Okay. This will be the end of the demonstration. Now that we've seen how ProtFlow works, we're hopefully all happy. And as a little bit of a summary of the tool chain that I showed in the beginning, everything is executed in this script except for the analysis. And an important concept that is there for ProtFlow is that you can switch up the runners that you have in here. You can switch up the order. You basically have full control that you have that is given to you with running this in Python. Okay. Another thing, since I only discussed 4 different tools, I wanted to show at least a list of all of the other tools that we currently have implemented. Of course, this is growing over time because new tools are always coming. But for now, we implemented really a lot of tools that we frequently use in the lab. As the next point that I wanted to discuss, I wanted to show a general conceptual overview of how ProtFlow actually works. And for this, I'll go over the main types of classes that we've shown. The first one is this poses class, which contains your proteins. This is basically a wrapper around the pandas data frame. So it has this pandas data frame to store all of the information that you need from your calculations. And it additionally stores information in the poses object of, for example, where is your working directory? Maybe you want a default JobStarter for your poses object and other things. Additionally, also this poses object allows you to do filtering, calculating scores, and some other things. Okay, and now when you want to understand how the function actually works, like how are we executing runners and everything, typically you pass your poses object to a runner. This runner is basically a wrapper around the design tool of your choice. And with the .run call, you activate this design tool. The design tool then compiles a list of commands. And this list of commands is passed to the JobStarter object. And the JobStarter objects are basically also their own class that have a function that they run that is called the start method that basically only just execute these commands, waits for them to be finished. And after finishing, sends them back to the runner. The runner then collects all of the scores and the outputs and sends them back into the proteins. And then you can directly execute your next runner if you want to. Okay. This comes with some benefits, this type of architecture, which is that if you want to use different runners, you can just switch them. And if you want to implement your new tool, you don't need to mess with anything about poses or job starters. You can directly just implement a runner for your tool and use it in ProtFlow. Implementation of a runner, to be fully honest, can take some time also for us, because you do need to automate all of this work that you would usually do if you didn't have ProtFlow, if you didn't have anything to automate it. And so we try to keep the workflow of a runner as predictable and simple as possible, which contains, as a first step, you always need to set up a working directory, because we want to keep the output structure of ProtFlow runners standardized. So it should always be in the working directory of your current poses object, and the output directory of your runner should always be named after the prefix that you give the run. And this way you always know where your outputs are, as also the guys from Ovo discussed. The next thing is that oftentimes protein design pipelines crash, or maybe you filtered out everything that you ever designed and also then you crash because you don't have any proteins anymore. And for this reason, you often need to restart pipelines, which is why we implemented that existing outputs are just skipped over unless you explicitly tell ProtFlow to redo the calculations. So there's always the override option. Then after this, we compile the commands, and these commands are sent to the JobStarter. And by isolating this job execution, you really don't need to worry about how you're going to run your tool on the system. This just needs to be, the JobStarter needs to be an object that can execute commands, and your runner just needs to compile these commands that will be executed. And as the last step, of course, the outputs of the protein design tool are collected and returned into the poses object. And we also have standardized runner output object for that. So you can always check how it should look like when you want to return your runner output to the poses. Okay. And this discussion of the JobStarter brings me directly to a question that maybe some of you had. How does it work if I want to make a new JobStarter, or maybe which JobStarters are even available that I can use? Maybe the thing that I should say here is that for ProtFlow, we implemented two very general purpose JobStarters that we basically use all the time. We don't need an additional one. The first one is the local JobStarter, which runs with the Python subprocess module. This means that you should be able to run ProtFlow scripts with this JobStarter on anything that that can execute stuff with the subprocess module. So most Linux environments, Jupyter notebooks, and also in Google Colab recently. And the second one is an SBatch-ary JobStarter. This is for when we want to move our Python pipelines or ProtFlow pipelines onto computing clusters. This is basically just for the Slurm scheduler. Can also schedule everything in parallel, just to run everything with SBatch job arrays. Okay, and in case this was not enough for you and you have something very specific, a very specific way to start jobs on your system, here's how job starters are built. We try to keep them as simple as possible on purpose, because this is the most likely thing that you will need to write if you have to. And it's basically a class that needs this method to start commands. This will receive from every runner, it will receive a list of commands that you need to execute. You need to wait for the completion of these commands and then just send a signal to the runner that you're finished. And as long as you can implement this within this class, you can always implement a new JobStarter. Okay. Going back then to the overall overview, some of you may wonder, when you set up this entire ecosystem of ProtFlow that is supposed to run tools, how does ProtFlow actually know where the design tools are living that you want to use? So if the runner needs to compile a list of commands to run a design tool, it needs to know where your design tool is in order to run it. And all of this is done with a single file. This is the config file. As a schematic here, this config file just points to where your tools are and where the Python environments for these tools are. In plain code, here's like an excerpt of how this config file starts. And of course, it continues downwards, where we already predefine the variables that you need to fill in if you want to use this tool. So for example, only if you want to use ligand MPNN do you need to fill in these variables, where you just search for your ligand MPNN installation and you add this path to the run.py script of ligand MPNN, and then the same for the Python environment that you install to run it. And with this, you should be able to run it. Okay, and a good thing about the system of config files is if you have a large lab where maybe everyone installs a different tool, or you have one person that installs all of the tools and everyone else is supposed to use these installed tools, then you can just write one config file, and this config file can be shared across the entire cluster as long as you are in the same user group and have permissions to access this file. But it's something that I do a lot with my students. They don't need necessarily to install all of the tools. I just install, or we just install ProtFlow, and we link my config file into their ProtFlow, and then they can just start running their script. Okay. And with this all explained, I hope maybe I convinced some of you to actually give this a try and give you a view of how to get started with ProtFlow. So first things first, we tried to write a documentation as much as possible that is documented with docs. This documentation is linked in the GitHub repository. So on the top right, you can just click and you will directly get linked to this documentation, where we have a quick start section that includes how to install ProtFlow, how to configure your first pipeline. And then it also contains some real world examples in the tutorial section, or let's say for now, just one real world example of a binder design pipeline that includes kind of all of also the dirty work that we have to do when making a design pipeline work, which includes RMSD calculation, really complicated filtering, and plotting all kinds of stuff before you know how to go on, and things like that. And this is from an actual binder design pipeline that produced binders. This was written, which is a fun side note, this was written by Adrian and Sigrid as a project to teach Sigrid how to write or how to use ProtFlow. And the binder design pipeline that they used and the binders that they designed, they eventually submitted to the adaptive binder competition, to the first round. And this one design that was submitted was then the one that made the third place, submitted by Adrian. If you want to know a little peek into the details, you can also check out this specific binder design tutorial. And then as a last thing, we also try to document all of the tools. But of course, ProtFlow is very big now, and this is very much a work in progress. So we kind of just started with this part of the documentation. So this is documentation. We didn't stop there. Horst made a very great effort when he tried to show people at a larger workshop how to use ProtFlow. He actually made ProtFlow work in Google Colab. And so you can also check out how to run a short binder design pipeline in Google Colab, which will show you how to instantly set up the config file, install like the most necessary tools, and it gives you kind of an idea of what you can do in there as well. Okay, and of course there are also Jupyter notebooks that you can run in the example section of the ProtFlow repository. In case you already started, or maybe you tried it and you really liked it, Adrian and I are very open for contributions for the community. This tool is in its very, very early stages, it's basically in baby shoes. And we really need user feedback, basically, to know which parts of our workflow library are difficult to understand and which parts you really like, which we could extend, maybe also which features you want us to implement. And just if you want to contribute, we want to note that ProtFlow currently is version controlled with Git. We develop on multiple branches. We set up already automated build testing with GitHub workflows. And of course, at this point, the repository is maintained by Adrian and by me. Okay, and we don't stop at this part. We also have a roadmap for the future. One of the more important aspects of this roadmap came out also of the European RosettaCon this year, where a lot of people said that installation is really one of the biggest problems. And so support for Docker and Apptainer is something that we really want to get going in ProtFlow, but yeah, we just need more hands at this point. And of course, also tool documentation to make the tool much more usable, and eventually integration of ProtFlow into pip and conda to make the install easier as well. And with this, I'll conclude my presentation, and I think we can go directly into the questions.
Great, thank you very much. Another round of applause and virtual applause. I think that was very cool, and I'm super happy now that you presented ProtFlow. I'll start with the same question that I sort of know the answer already, but here, how hard is it here to wrap a new tool? That basically means you have to design a new runner, right? So you have to parse the outputs, and that's sort of it. But that sort of seems like the minimal amount of work to wrap a new tool that's even possible.
Yeah, so very important question. Depends on the tool. Something like Omega Fold, I would probably need a day of coding, and then you can use it forever. And bolts, I recently integrated with special options parameters, so like special classes to make the handling easier. This implementing bolts, including a tool documentation and everything, took me a week. But again, once the tool is integrated, you can basically use it forever.
Very cool. Then do we have any other questions? if you have any questions, please feel to raise your hand or type them in the chat. I'll also give a comment. I think you already support Dockers right now because you can call obtainers directly. So you can just pass the command line string to call the obtainer with the correct obtainer run or execute in your config file, and things just work. Because this is exactly what we're doing in ProtFlow, and it really makes it easy to install. And if you look at the ProtFlow website right now, it's just installing 5 containers, and then in the config file we just place the full command line that includes the paths to the container, and then the, I don't know, RF diffusion or whatever, and things just work. So I think you've got it down already.
That will be great. Thanks for the notes. Maybe I'll check it out or ask you also.
I can send you the link, and we can do this offline on Slack. Yeah.
Thanks.
Then any other questions? We have a question on how fast is the pipeline in terms of processing output speed, but that depends on your hardware and the programs that you're using and the size of your proteins and stuff like that. So
yeah, but file size as well. So for example, we also developed Driftive, and Driftive in some cases designed like 40,000 sequences or more than 10,000 sequences, depending on how your run looks like. And then it can take several minutes to read all of the output scores, and your data frame can also get like several gigabytes, so maybe tens or hundreds of gigabytes large. So for really, really large design runs, you want to calculate that this takes maybe a few minutes. It's not instantaneous, but you want to run on a high performance computing cluster anyways, because predicting 20,000 sequences on a computer with one GPU will take a week at least.
That's very cool. I guess the only assumption is that you have a shared network file system, right? Because the runners and all the locations are sort of absolute paths on some sort of mounted NFS.
Yeah. Yeah, if you want to use tools that other people installed, of course, you need to be able to access these other people's users. But yeah, if you're in the same user group, then you have these permissions. If that answers the question.
Very cool. Are there any other questions?
Quick question. You can turn on the mic. Thanks, Marcus. Cool to see the overview. I'm playing around with Driftive these days, so really cool pipeline. I think a very smart way to use RF diffusion. I was wondering, for that pipeline, could you share some example design results? I would love to kind of build some import logic for OVO that you could use it. You could use OVO just to visualize the results. This pipeline.
Yeah. Sorry for not responding to your GitHub issue. Yeah. So for Driftive, every section of the pipeline, so the initial screening, the backbone refinement, the evaluation, and the others, they produce folders that are called screening_results, refinement_results, and so on. And in these folders are the filtered outputs of these stages, and including a PyMol visualization script that you can run. Should be called align_results.pml. And as long as you're in that same folder, or like you have that same folder in tag, you should be able to run this with PyMol and open everything. And also the scores of your proteins at this stage should be in a JSON file in this results folder or in the folder beneath it. Also, please contact me by email if you have any more questions on this. I'm more responsive there, definitely.
Cool. So another round of applause for our speakers. Thank you very much. This is very cool. And I hope you enjoyed this protein design workflow meeting. I definitely did. Great. Well, take care, everybody. Bye.